This is the second part in the microbial growth and culturing methods video series. In the previous video, we talked about environmental requirements for growth. In this video, we're going to be talking about media, which is going to be used to grow microbes. In another video, which was microbial identification techniques, we talked about some aspects of media. However, in this video, we want to be able to go further into it and to explain some other aspects, including its form, its functions, and its ingredients. Media can come in two different forms. It can come in either a liquid form or a solid form. If it is a solid form, we call it an agar, and if it's liquid, we call it broth. The next thing I like to classify as the function. The true function of media is to grow bacteria. However, there's some specialized function that some media have, and that's going to be our selective, our differential, and our reducing. So again, our selective media is going to select for one, either a single microbe or a group of microbes, and it's going to suppress the others so that they're not going to grow. So this has something in the environment that's going to allow one to continue to grow while the others will not be able to grow. Differential media is going to allow us to differentiate, or in other words, see differences. So differential media allows you to see differences, and usually it is by color. As a reminder, in differential media, when you have these differences seen, a lot of times it is taking advantage of a metabolic process, or in other words, some microbes can break down something while others cannot. The product or the byproduct of them breaking down something usually leads to a change in the pH, and the change in the pH causes an indicator to change a color, and that's usually what is happening with differential media. The last one is reducing, and this eliminates oxygen. The reason why it's really beneficial to eliminate oxygen is because oxygen is going to be necessary for aerobes or aerobic organisms, but if we have an obligate anaerobe, we cannot have any oxygen. So therefore, we need a reducing media, because if it just used a regular medium, we would not be able to grow the obligate anaerobe. In addition, you can also see whether or not something is aerobic, anaerobic, or facultative anaerobic based upon whether or not it does or does not grow with the reducing media. So again, these are the three things that I like to define as the functions, because these are specific functions that some media have. The last aspect that I want to talk about are the ingredients. Ingredients in a media can either be complex or chemically defined. In a complex media, the ingredients are very generalized. In other words, you could break them down further and be able to get more out of them. An example of looking at this is a cake. A cake calls for many different things. It either calls for flour, for butter, for milk, for sugar, but if you took those things, you could break them down further. So for example, milk isn't something that just exists on its own. It can have fats, sugars, and proteins that can be found in that. And so therefore, the ingredients used to make a cake are very generalized. The same thing is happening with these media. The great examples that you'll see are blood, beef, yeast, and if it says something, extract. A lot of times you see beef extract or yeast extract. So those are your indicators that this is a complex media. If you see those on any of your assignments, just really think, okay, that must mean it's a complex media. Chemically defined, on the other hand, are going to be very defined. So these are very specific. These pretty much look crazy because they're going to look just like elements. These usually are chemical compounds, therefore. That's what I will kind of have as your example. So instead of saying salt, it would actually say sodium chloride, like NaCl. And the reason why they're so defined is because you couldn't break them down further until they're down to their actual elemental state. And so these chemically defined media are going to look very, specific. And remember that these two are describing the ingredients. I want to make some notes on complex and chemically defined, things that students miss often. These are describing the ingredient, not the amount of the ingredient. So just because you might see something that says 0.05% yeast extract, that is still a complex media. My advice to you is that as you are looking through things, cover up the amount so that it does not distract you. Look at just the ingredient. If you see that ingredient is complex, therefore it's complex. If you see that they're all very specific, it's going to be chemically defined. Another note that you should make is that if one ingredient is complex, the entire thing is going to be complex. So though you might see a ton of chemical compounds and then you see one yeast extract or one beef extract ingredient, that means it automatically is complex. So again, just make sure that you don't look at the ingredient amount, look at the ingredient itself, and then make sure that you remember that even if one thing is complex, the entire medium is complex. So these are the different aspects that are with media, with its form, its function, and its ingredients. I'm going to erase the board now, so if you have not completely written this down, I would recommend that you pause the video now and do so. The last aspect that I want to talk about now is actually putting your bacteria on the medium. So there are two major methods that are used, although there are many more that you can discover if you decide to look on further. The two main methods are the streak plate and the pour plate. First, I want to make mention that if you are in the lab class, you will do the streak plate and the pour plate method. However, if you're either in an online course and not on campus, or if you are not in the lab class, I will explain how these are done. The streak plate, you're going to pull from a culture. So you're going to have a vial of bacteria already for you. And you're going to take what we call an inoculating loop. So basically it's just a metal loop. And you're going to dip it in here, and then you're going to streak it onto a plate. So you're going to streak, then you're going to pull across and streak, you're going to pull across and streak. And you only put it in once, and in between each one, you just pull without re-dipping. And then you're going to incubate it, and this is kind of what you're going to see, and this is the hope. The first one is going to have a lot of bacteria on it because you pulled it straight from the culture. So you're going to see a lot of bacterial growth here. Then you're going to see some bacterial growth here with maybe some isolated colonies. And then here, you're only going to get the isolated colonies. These are what you're looking for because we call those pure colonies. And that's what you're able to isolate and to do identification techniques on. The other method, the pour plate, you're doing exactly what it sounds like you're doing. You're literally pouring the bacteria onto the plate. So what you do, you have multiple tubes, and you're going to dilute them. So you pull from the main culture into the first, then from the first to the second, second to third, and you can do this multiple times depending on how many dilutions you want to observe. And then you're going to put it on to a plate, and you're just going to pour it. And the hope is that as you grow, you're going to see the isolated colonies. And so because you poured, it evenly distributes it. And so these are the two methods through which you can put bacteria onto a plate. So in the next video, I want to discuss how we're going to observe the growth.